Well, thank you very much, Chris. It's a great pleasure to be here. As Chris sort of intimated, I'm a biologist. I'm a biologist who has a passion about insects. I began my life as an entomologist from a very young age. It's taken me on a very strange journey, and I'm going to take you on that journey today. But I want to step back and give you a big picture view of current biology, what biology is as a discipline today. Now, to understand what we're about in biology at the moment, we need to consider the major advances, if you like, or paradigm shifts, as they're sometimes called, in biological thought over the years. Now, perhaps the first revolutionary in the study of biology was Linnaeus, because it was he, in 1735, who published the first account of how we might go about categorizing, cataloguing the incredible diversity of life on this planet. So it's he who devised the system which is known as binomial nomenclature, the way that we name things scientifically such that local names, which differ from place to place, just don't cause confusion if everybody uses the same scientific nomenclature. Then along came Darwin, who provided a theory which explained that biodiversity. So we had a way of naming it thanks to Linnaeus, and now we had a way of explaining it thanks to Darwinian natural selection. And his very famous book, which quite literally changed the world, was published in 1859. In fact, as Chris said, when I was at the University of Oxford in the Museum of Natural History, it was in that building that there was the very famous debate over Darwin's theory of natural selection between Huxley and Bishop Wilberforce, the Bishop of Oxford. Then, along, particularly in the 20th century, came a whole series of scientists who put together Darwin's ideas with the new science of genetics. Darwin knew nothing about genetics. He had no mechanism for natural selection. Mendel and subsequent geneticists provided that mechanism, and the neo-Darwinists put it together. They put those two things together, and it was called the neo-Darwinian synthesis. That took up much of the latter part of the 20th century, and it's still a vigorous discipline. We then, thanks to Watson and Crick, had the mechanisms of molecular transmission. So we had an understanding of molecular biology. The molecular revolution commenced as a result of their famous description of the structure of DNA, and it continues at a great pace today. So molecular biology has revolutionized the way we think about biological systems. And I would argue that we're currently in the fifth great revolution, and I'm going to call that the epigenetic revolution. And it stems from the fact that even though we, for example, know the full sequence of the human genome, that has turned out not to provide us with an understanding of human beings. The reason is that just having the genetic code is not enough. We need to know how genes interact with one another, how they interact not only with other genes but with the developing environment within the embryo, and then throughout the life of the organism with the actual outside environment. So, as I say here, epigenetics is the focus on how adaptive phenotypes, and by a phenotype I mean the way that animals are and function, or plants or indeed other organisms, and how those adaptive phenotypes arise during development as a result of plastic interactions between genes and the environment. Now that's helping us increasingly to understand how genes play out through development to yield the way that organisms are, the way they work, the way they survive and succeed in their environments. But the final frontier, and this is what I'm going to try and focus on today, is to take that scheme even further, from genes ultimately to ecosystems. We live within a complex biological and abiotic environment. How ultimately does that work? Now, what we know, of course, is that genes, through development, yield phenotypes, organisms. Those organisms interact with one another to produce population-level responses. Populations, then, assemblages of different species, contribute to make up communities of organisms, and they in turn produce complex ecosystems. But it isn't a simple one-way line where you go from genes to phenotypes to populations to communities to ecosystems. The whole thing is girded by abundant feedbacks. So you can take any one of those boxes and you'll find feedbacks to earlier boxes. The whole thing is a complex, interconnected network. Sitting at the heart of that is the phenotype, the way organisms are. And it's that that's seen by natural selection. Natural selection is the engine that drives this whole set of processes. Now, that's the background. What I'm going to do now is take you to North Africa. And I'm going to begin with a story which I said initially is a strange journey. It's my journey. It's a journey that's taken me a little bit through those interconnected networks that I showed you just in the previous slide. And it's led me to consider the human adaptive phenotype as much as that of this animal here. So, this is a shot or a film taken in the midst of a swarm of the desert locust in North Africa. Now, this animal, along with about 20 other species of grasshopper, is defined as being a locust by possessing one peculiar piece of biology. It's not a normal grasshopper because it's effectively two animals packed within the same genome. Now, to illustrate what I mean here, these two are sisters. They were sisters from the same batch of eggs. The one on the left, the beautiful green grasshopper here, was reared on her own. The one on the right was reared in a crowd throughout her life. Now, these are the last larval stage before at the next molt they develop wings and become winged adults. But what you can see here is that these two things are really remarkably different. And in fact, until the beginning of the 20th century, they were classified as being different species. They're so different. So Linnaeus was the first to give this animal its scientific name, but he gave it two different scientific names, and it wasn't until a Russian biologist called Boris Uvarov appreciated that they're in fact the same organism, just reared under different conditions of crowding, that they were put together into the same species. So, the one on the left is called Phase solitaria, and what you can see if you look at her is that she's green and hence in amongst vegetation would be well camouflaged. If you put her near other locusts, except when she's adult and reproductive and needing to mate, she would avoid them. She would be very cryptic in her behaviour. She would freeze if she saw movement, she would creep away from other locusts, and she would live a solitary life. In contrast, the animal on the right is much more brightly coloured, yellow, black, and white. She would be actively attracted by other locusts, would aggregate with them, and if there was sufficient local numbers, would start marching and produce these mass marching hordes, which then move through the environment, eating everything as they go. And when they become winged adults, they take off in huge swarms, which can cover hundreds of kilometres a day. They can number trillions of insects. And you hear these staggering statistics, such as a single swarm of these things in one day can eat the same amount of food as the entire population of Australia can in a week. So they're individually quite refined eaters, but en masse they cause devastation. Now, the really exciting thing about this weird transition, these two animals exist within the same genome. So the reason that this one grew up looking like that rather than that was purely because of its environment. It grew up on its own. This one grew up in a crowd. That is the heart, not only of a really interesting piece of biology, it's also the heart of the locust problem, because if they didn't do this, if they didn't shift from that form to that form, they wouldn't be a problem. They wouldn't be a pest animal that affects the livelihoods of 1 in 10 people on the planet. Now, you'll see here a whole range of different so-called phenotypic traits which differ between these, their appearance, their shape, and so on. But the thing that changes first, if you put one in a crowd, if you put this one in a crowd, for example, is its behaviour. So quite literally, if you crowded her, within a matter of a couple of hours, she would be behaving as if she was in that form. She wouldn't look like that yet, that takes several molts to achieve, but she would be attracted by others, she would be more active, she would contribute to a marching band, and that only takes a couple of hours. So one of the key things we wanted to know at the outset was, what is it about being in a crowd that can cause this animal to change to this animal quite so quickly in its behaviour? Well, let's have a think about that. What are the potential stimuli that might be associated with crowding that cause that transition? Well, it could be seeing other locusts. It could be smelling other locusts. It could be being jostled by other locusts, contact, in other words, with others. Or it could be the smell of other locusts, the sound of other locusts. Well, to cut a very long story short, we teased these 4 sources of potential stimuli apart in a set of experiments and found that the critical stimulus is touch. So physical jostling can cause that change in behaviour within a matter of hours. If you jostle that green insect within 2 to 3 hours, she will be behaving as if she's been reared throughout her life in a crowd. Now, physical contact among insects involves them detecting being touched. But insects are covered with touch-sensitive hairs, like you are. You have touch sensitivity across your entire body. So that raises the question, does it matter where you touch another locust to get it to shift from its solitary to its gregarious form? Well, to answer that, we set about touching them in various places using a paintbrush. So this, I think, if nothing else, illustrates that science need not be extraordinarily difficult and technical. It just takes quite a lot of patience. I spent many, many hours sitting in a warm room, stroking locusts on one of 12 different body parts for 5 seconds a minute. And at the end of 4 hours, we could then assay the behaviour of the animal and see to what extent it had been changed. And then you can produce a sort of heat map of the body surface indicating where touching was effective in causing that change. Well, the answer was it was only effective if you stroked the hind leg. So, stroking the thigh of another locust, or of a locust, is sufficient to induce its transition from being a shy, cryptic animal to being a social, gregarious, party-going animal. Now, of course, you can try that with your friends in college. Preferably not here, no, it's not. Now that actually turned out to be a really important observation because it enabled us then to focus our attention on one part of the animal's body. If we wanted to try and then track those stimuli being touched on the leg ultimately to the translation into a change in the animal's behaviour, we had somewhere to start looking. In fact, what it did was it opened a window on the underlying neural and chemical processes of that change. The very next step was to identify the nerves that pass from different touch-sensitive hairs on the hind leg, and then to trace their pattern of input back into the central nervous system. Now, insects have a whole series of brains in their body. They have one big brain in their head, but then they have a whole series of local brains all the way along the rest of their body. The nearest local brain to the hind leg sits just about here in their chest. And what we found was that there are neurons that carry information from these touch-sensitive hairs that send their electrical impulses back into this part of the nervous system. And we were able to identify which were the critical inputs by doing a combination of snipping of various nerves and also implanting very fine wire electrodes into a free-moving animal and then electrically stimulating those nerves. We found that you could send patterned electrical stimulation into these nerves via these electrodes, and even when the animal had no contact with anybody else, you could induce the change in behaviour to the gregarious state. We then asked the question, all right, we've identified the key stimulus, which is touch, we've identified where it's effective, the hind leg, we've identified the neural pathways involved, but how do they cause a change in behaviour? Well, if you think about it, that change in behaviour has to occur, well, it does occur within a matter of hours. That means that whatever these signals are that are coming in and having their effect here cannot be causing complete physical rewiring of the nervous system. Behaviour is the product of the nervous system. The nervous system, in the case of locusts, comprises about 1 million nerve cells. There's 100 billion in our brains, but nevertheless, 1 million's many fewer, but nevertheless it's quite a large number. There's 1 million nerve cells in the total nervous system. There's probably about 10 to 20,000 in this one part of the nervous system. They can't be physically rewiring within that period. It's just too short. So what we supposed is that these impulses are causing the release of chemicals in the nervous system that are acting upon existing neural pathways and causing, if you like, the mood of the animal to change by affecting the strength of connection between nerves in that network of nerve cells here. Now, there's a whole range of potential neurochemicals that might be involved. The nervous system of this animal, and indeed ourselves, is a soup of chemicals that float around, acting on receptors, membrane-bound receptors within the nervous system, changing the activity of individual nerve cells, changing the strength of connection between nerve cells and neural pathways. So we went about trying to establish the time course of change in that part of the nervous system of a whole range of different neuromodulatory compounds. One stood out as being, of showing the right time course as the behaviour. So there was one really key candidate compound. So then we had to go about manipulating levels of that compound in the nervous system by using a whole series of different drug treatments, so pharmacological manipulations where you block receptors or you stimulate receptors or you cause production of the compound to be toned down or pushed up. And in every single case we found the effect on behaviour. So this one compound was both necessary and sufficient to cause the change in behaviour upon activation of these pathways that stimulate or that indicate crowding. What was that compound? That compound was serotonin. Now, serotonin is found in your nervous system as well. It's found in the nervous systems of most animals, And in every single case, it seems to play a role in arousal, sociability, and other associated sorts of behaviours. It, of course, you probably know, is the source of manipulation when it comes to the treatment of depressive disorders. So one of the major pathways that's manipulated in the treatment of depression is the serotonergic pathways in the brain. It's also the source of manipulation in some of the recreational drugs that are taken by young people these days. So ecstasy, for example, acts upon this same pathway. Now, that, you can imagine, caused quite a lot of interest amongst the press. So we had plagues of locusts and ecstasy, we had bugs on drugs, and we had all manner of other things. So if you look on the internet, you'll find endless versions of this in the world's press. All right, so what we then had was a behavioural change which sits at the heart of the locust problem. We'd asked the question, what is it about being in a crowd which causes that behavioural change? We identified where the source of stimuli are. We traced the pathways back into the nervous system, and we'd ended up with a key chemical, the release of which, in a burst upon initially being stimulated by being in a crowd, triggers the whole process of change. What I want to do now is to say, all right, we can now understand why an individual might change in relation to being crowded, but why did they come together in the first place? So solitary locusts, I've already told you, avoid one another. They don't like going near one another. If they happen to be brought together, then they'll jostle and you'll get everything that I've just told you about, which will flip them into finding others attractive, and then they aggregate, and as we shall see, that can roll upwards in a positive feedback like a snowball. But it doesn't answer the question, why do they come together in the first place? Because in the wild, they're not put together in boxes in the lab like I did, or they're certainly not put down and tickled with paintbrushes. So we need some explanation for how they're brought together against their predisposition to avoid one another. Well, the answer to that came from using computer simulations to simulate real locusts, or at least locusts that we program to behave according to the way we know real ones do, and to allow them to roam freely within a simulated environment where we manipulated the number of locusts and the amount of food and the distribution of that food, how clumpily it was distributed. And what we found from those models, which then allowed us to go and do experiments in the lab and in the field in North Africa, was that the critical thing is how clumpily distributed food is in relation to the numbers of locusts present. So if you have a certain number of locusts in the environment and they're able to avoid one another, they will. But if the food becomes more and more concentrated, then they have to go there to feed, whereupon they'll encounter other locusts, they'll jostle one another, and then they get flipped into the gregarious behavioural stage. So that's the mechanism, and we were able to use computer simulation to explore that in conjunction with, in dialogue with, experiment, both in the lab and the field. We also wanted to know, well, what's the reason for doing this? What's the function? What's the evolutionary significance? That's the sort of question Darwin would have asked. And we've found, again, using mathematical models, that there are various possible explanations. Some of them simply to do with being safe in numbers. If there's a large number of you in an environment, you're safer in an aggregation than if you remain pretending you're not there in isolation. Although, as we shall see soon, there's a risk to getting together with your colleagues in large numbers. The other is that we were able to show that if you're totally aggregated in a sea of otherwise emptiness, you're harder to find. You disrupt the pathway of foraging of some of your predators. And that's a similar idea to in the Second World War, when the North Atlantic boat convoys were trying to avoid the U-boat attacks from the German Navy. They did so by ganging together in little groups, and a small cluster of boats in the vastness of the North Atlantic is harder to locate than a series of separated boats. So again, there's a series of mathematical models that you can build and test in relation to both simulations and the real biology to try and understand why as well as how things happen. Two of the big questions in all of science is why something happens and how something happens. All right, so we've got the environment encouraging scattered locust populations to get brought together so that they then interact with one another. That causes them to flip into the gregarious behavioural state, which will produce local clusters of locusts in the environment. So what you end up with is jostling aggregations of locusts going nowhere in particular, but then suddenly, as if of a single mind, the whole lot will start to march. And they march in a highly aligned, cohesive way, like an army, moving through the environment. Or a river of particles is another way of putting it. So you've got this advancing front with a tail of locusts forming this moving aggregation, which can be hundreds of meters across, it can be certainly hundreds of meters long, and contain billions of insects. And these are juveniles, they're not yet winged, and they're hopping along, walking side by side with one another. Why do they do that? Why do they suddenly decide, in inverted commas, to start moving collectively? And that transition is quite literally as rapid as the transition of water turning to ice at an appropriate temperature. It's a phase transition in the mathematical and physical sense. Now, there's a couple of possibilities, of course. You might have leader locusts. You might have the equivalent of an army where you've got general locusts who say, start marching, and off they go. Or else it could emerge simply as a result of everybody doing their own thing, and collectively that produces what's known as an emergent outcome, in this case, collective movement. Well, to try and explore those possibilities, we turn to our statistical physics colleagues. Now, there's a whole bunch of physicists around the world who are working on a set of models called self-propelled particles models, where they're trying to explain the behaviour of collectives of particles and trying to explain them in terms of simple local rules that are employed by individual particles. So what you do is you take in, and this is a simulation, you take individual particles that behave according to some local interaction rule, you simulate large numbers of them in the computer, and you see whether the whole group does something interesting that you couldn't necessarily predict on the basis of what you programmed individuals to do. Well, using that approach, we demonstrated that in fact collective behaviour doesn't involve generals and leader locusts. It arises from simple local rules. And the basic rule is align with your moving neighbours. And locusts, and we know this because we've quantified it, respond only to neighbours within a diameter of about that. So it's about 13.5 or so centimetres radius. They will respond to anybody within that. So far as they're concerned, beyond that, nobody else exists. By following that simple rule, when my neighbours start to move, I will align with their mean direction and move in that direction. When you reach a critical density locally, the whole lot will start to move together. You get that phase transition. Now that was the mathematics. We needed to be able to study the behaviour of the computational theory with the behaviour of real animals. Now it's a challenge to start working in the lab with locusts that are marching together, because if they start marching and they go out the door and down the corridor, it alarms one's colleagues and it doesn't make you terribly popular in the department. So we needed some way of making them think, in inverted commas, that they were marching, but actually they were going nowhere. That's where this comes in. Now, it's not a fried egg with flies all over it, as you shall see. It's what we call our Mexican hat marching arena. So it's a circular arena with a dome in the middle, and the locusts will march round and round and round and round endlessly. And we're able to quantify their position and hence to derive their local rules of interaction, their degree of alignment within the group, and so forth. And in that way, we were able to show that the predictions of the theory, the statistical physics models of collective behaviour, was met very precisely by the behaviour of real locusts. Now, that was in the lab. We were able also to do the same thing in the field, and we've done that over the last couple of years in outback Australia, where there's another species of locust that does something rather similar. We were then able to go from the field, where we were able to measure some of these interactions by setting cameras up and watching locusts flow underneath. We're able to go back to simulation and simulate very large numbers of locusts and see whether we could produce the overall structure of a real locust band in the field. So what I'm going to show you now is a couple of simulations. Now, we're able to simulate quite literally millions of individual interacting agents using what's known as CUDA computing, which involves wiring up in parallel a series of graphics cards from gaming computers, a very cheap way to get a supercomputer. So what we have here, I'm going to show you, is 250,000 locusts simulated over 4 hours with certain behavioural rules that we've derived by recording what real locusts do, both in that marching arena and in the field. And we're going to see how they behave in simulation. So what we have here is those 250,000 locusts moving collectively, producing a band which has a crescent-shaped front with very high density at the front, and then this comet tail of decreasing density as you move further back. So the scale of this in the field would be several hundred meters across, and the time course of that would be substantially longer in real time, of course. And if you look at real locust bands, here's one from the air. There's the crescent front, that's 500 meters across here, and you've got the sort of tail of the comet back here. So this reflects quite closely the behaviour of this locust, the Australian plague locust. But different locusts around the world have very different and typical band structures. Their bands do very different things. And what we found was that by making very simple adjustments to the rules whereby locusts interact, making them, in this case, be slightly more strongly attracted than in the last, you can get the same set of locusts to produce a very different band structure. Now, this one here reflects very closely the pattern that you might find in the South African brown locust, which is another of the 20 or so species of locust. Its marching bands have these sort of filaments, individual little rivers and filaments that cross one another and move onwards collectively in a rather different way to the Australian plague locust. But what we can show is that by making just a simple adjustment, you can get a very different outcome at the level of the group. The next thing we need to do is to take those models and to go back into the field and to see whether they're able to predict very precisely how individuals will move with respect to one another in a real marching band. That's a problem, because if you've got 1 billion locusts and you release one here, you've got no idea where it's going to be 2 hours hence. And you need to know where that individual and this individual and that individual are to be able to test the predictions of the model precisely. To do that, we turn to some very talented engineers at the University of Sydney in the Field Robotics Laboratory. And they've built for us, in fact, it's not a helicopter, it's a fixed-wing aircraft, but they've built for us an aircraft which has an onboard stroboscope which sends out very bright flashes of light, which then reflect back off little crystals we mount on the back of our test locust that we release into the marching band. Now these things are so little, the locusts are small enough that you can't use sort of traditional radar techniques. You have to do something different. So we have a little crystal, like a little diamond, it's a glass crystal on the back of an individual. The stroboscope flash is reflected back from the crystal, detected by a detector, and then it's incorporated into a tracking software, and the plane will track autonomously. It's a robotic aircraft. It will track the movement of the swarm over time. So in that way, we can track how individuals, up to 20 or 50 individuals that we release in a particular place, in a particular configuration, move and disperse over time, and we can use that to derive very precise data to test our models. And in this way, we're able to predict better where locust plagues will move. We can then advise the control agencies how better to treat them in a more efficient manner with insecticides, and perhaps also to be able to use biocontrol agents, which take a little longer to act than a traditional chemical poison, earlier in the cycle of outbreak, and hence prevent many of these problems. And we're currently working with the United Nations in North Africa to help implement that, and also with the Australian Plague Locust Commission here in Australia. Still leaves a question, however. Why do locusts align with their moving neighbours? Well, the answer to that question turned out to be rather sinister, and it came from another mass-marching grasshopper-type creature that lives in northwest USA. It's a thing called the Mormon cricket. This is a band of Mormon crickets. Now, Mormon crickets are about as big as your thumb. They're wingless. They're related to locusts, but they're not locusts. And they form these vast marching aggregations, marching bands. This one, we filmed crossing that road in Utah for 5 days. It travels about 1 to 2 kilometres a day. So it just gives you an impression for the size of the numbers of animals, the sheer weight of numbers of animals there. This animal doesn't strip the environment bare as it goes through, unlike locusts, which tend to eat pretty much everything in their path. And it wasn't clear whether they're marching simply through lack of food or whether there was something more precise involved, some more specific deficit in their habitat, which was encouraging the whole thing to keep moving. So we set about trying to understand what it is about their environment. This is their environment. This is the field site where we were working in Utah, which caused or was associated with this mass marching phenomenon. So we went out and we spent a couple of weeks in this particular experiment, which was the best fun in the field I've had for many years. It was terrific fun. Field work is a lot of fun, I must say. And what we did was to ask the locusts what they, or not the locusts, the crickets, what they wanted from their environment. Now this is a guy called Ian Cousin, who is working in my lab in Oxford and is currently on faculty at Princeton. He's now become a really quite famous collective behaviour biologist and simulator. And that's another guy called Pat Lorch from Kent State University in the US, and that's me protecting myself from the sun. Anyway, what we did was we placed in front of marching bands of Mormon crickets 4 different sorts of dry granular food, which we could manipulate, within which we could manipulate the protein and carbohydrate quality. Those are the 2 major macronutrients that these animals, and indeed most animals, require from their diet. Fat in their case is a relatively minor component because they typically eat vegetation, but not always, as we'll see. So we thought, well, let's see whether protein and carbohydrate, or either of those two, is associated with their collective movement. And the logic was that if we put down dishes of these foods in front of them and saw their likelihood of stopping and eating, then that would give us a clue. It's running a census, if you like, of the animals. Which one of those four do you want, if any? So these four were either a food that contained both protein and carbohydrate at 21% dry weight, which we knew from studies on locusts is about the optimal dietary composition. One that contained the carbohydrate only in 42%, the sum of those two, but no protein. So this is a carb-only diet. This is a diet containing only protein but no carbs. That's the Atkins diet for a Mormon cricket, if you like. And this one here contains no protein nor carbohydrate, just the cellulose and salts and fats and vitamins and other stuff that's present in all four. So we laid those four diets out, we allowed the crickets to come and contact them and watch what happens. And so too can you. Now when you're watching this, you need to bear in mind that Mormon crickets, like other insects, taste with their feet. They also taste with their antennae and their mouth, external mouth parts, their palps. So they can make decisions about nutritional quality without having to stop and eat something. I want you to watch this over the coming minute or so. And when you think you can see what's happening, somebody yell out what's going on. Right. Perhaps not attracted by, they're arrested by protein. So the probability of hitting any of those four dishes is the same, but their likelihood of stopping and eating is very much greater when the food contains protein. And you'll notice that when they contact carbohydrate only, they touch it and they keep moving. The zero diet, they just don't stop and eat. But these two, you can see increasingly, they're glomming on in larger and larger numbers. And in fact, if you repeat this many times in different bands and different places, you find that indeed the probability of stopping at a dish is a function of the protein content, and they don't care whether it's got carbohydrate. There's no difference between the zero diet and the one that's only carbohydrate. So, protein. Their environment is deficient in protein. What's the most abundant source of protein in a Mormon cricket band? Other Mormon crickets, precisely. So the crickets are on a forced march to find protein, and if they stop moving, they get cannibalized. Now, this is a pretty extraordinary piece of cannibalism. In fact, I can find no other example where an animal takes as big a meal, an animal at least that chews its food rather than swallows it whole. A Mormon cricket can eat another Mormon cricket of the same size in its entirety within a couple of minutes. So you'll find that entire Mormon cricket now packed within the foregut of its cannibal within a matter of minutes. Cannibalism is the driving force. Cannibalism is the reason why Mormon crickets and transpires locusts align with their moving neighbours. If you don't align with your moving neighbours, then you're in trouble because they'll eat you. And indeed, in the case of Mormon crickets, they'll eat other things as well. If you were to fall down and be unable to move, I reckon you'd be a skeleton within a few hours. So this was a jackrabbit. It was roadkill, but its ears were somewhere out here half an hour earlier, and the crickets have gone for the soft bits and eaten its ears. And we did a whole series of experiments and showed that locusts, they're less cannibalistic than the Mormon crickets, which are fearsome cannibals, but nevertheless, cannibalism is a driving motive, if you like, for alignment and hence mass movement in locust swarms. And this guy, literally, Guy Smith, who you've probably heard of, he was the author of The Night of the Crabs, apparently. He published this book. He clearly had it all sewn up way before we did. Anyway. Now, we've come to a segue point in my narrative. Protein is the key reason why Mormon crickets cannibalize, and hence Mormon crickets and locusts mass migrate. In those experiments with Mormon crickets and in similar ones in the lab with locusts, we found that if you satiate them specifically with protein, they don't cannibalize, and as a result, they don't mass migrate. They have a separate appetite for protein, separate from appetites for carbohydrate and for fat. And you can manipulate those independently of one another. Now that turned out to be a really important discovery, because in most of the study of nutrition, it's obviously accepted that animals need many, many different nutrients, micronutrients as well as fat, carbohydrate, protein, if they're to thrive and prosper. But most of the description and particularly the experimentation and theory in nutritional biology is predicated on the idea that animals count calories. So we worry about the number of calories we eat. Calories, of course, are the products of nutrients. They're not the nutrients themselves. And what we showed here was that locusts have a separate appetite for different nutrients. Is that perhaps something which can help us in understanding our own modern human dilemma in the case of nutrition and obesity? Well, to try and explore that idea, I need to introduce you to a simple graphical way of considering the idea that animals require more than one nutrient at a time. So, if we go back to our locust example, locusts require a certain amount of protein to be eaten over a day and a certain amount of carbohydrate to be eaten over a day if they're to perform optimally, to grow best, to go on and reproduce as adults. You can think of that requirement for both nutrients as a target. The animal, if you like, is aiming at that target. It's not doing so through any conscious means. It's just designed as a result of natural selection to behave in a manner which will bring it to that point if it has the opportunity. So natural selection and evolution has led to animals that have particular physiological control systems which regulate their intake of these two nutrient groups to achieve that target if they can. Now, locusts, we found, are remarkably good at doing that. So, if you, for example, provide them with different imbalanced food combinations which allow them to mix a diet that gets them to that point, then they will do so. If you dilute their food 7-fold with cellulose, which they can't digest, they'll eat 7 times more to get to that same point. They have a remarkably powerful capacity or effective capacity to regulate their intake of protein and carbohydrate. We also knew, because we'd worked it out in experiments, how they managed to do that. So, for example, we'd shown that their protein status is reflected in their sensitivity of their taste receptors to the presence of protein in food. When they need protein, they have receptors that are very responsive to the taste of protein. When they need carbohydrate, their taste receptors are very responsive to the taste of carbohydrate. And there's a really elegant feedback there, which means that the animal tastes what it needs and eats what it tastes. But anyway, what happens if you don't allow the animal to get to this point? And as we saw, that's what happened in the Mormon cricket example. They were confined to having too little protein in their environment because there were so many of them. They picked out the high protein resources because the environment just has low protein anyway because of where it is. Now that's the equivalent, if you think about it, of not being able to get to this point because your environment isn't nutritionally balanced. It's pushing you off in some other direction. In this case, too little protein in the environment is a little bit like being stuck on one of these lines, which is taking you further up in the carbohydrate dimension per mouthful than it is in the protein dimension per mouthful. So we wanted to know what happens to locusts under those circumstances. How do they square that circle? If they can't get to here, what do they do? Well, you had to measure what they did, and to do that we confined locusts to one of a whole range of different foods that contained too little protein relative to carbohydrate, and measured how much food and hence how much nutrients they ate. They're these blue dots. We did the same with foods that contain too high a percent protein relative to that optimal point, and they're the blue dots. That's where the locusts travelled to and stopped. Now what you can see, that forms an array, and if you join those dots, the shape of that array describes the way in which the animal balances eating too much of one nutrient and too little of another when it can't get to its optimal intake, its intake target. And what you'll see in simple terms is that locusts were willing to overeat carbohydrate on low percent protein diets to a greater degree than they were willing to overeat protein on high protein diets. So you look at the length of that arrow. These animals would continue to eat and eat and eat and eat and eat until they got to that point, whereupon they've almost got to the right amount, the target amount of protein, but they've eaten hugely too much carbohydrate to do so. Whereas in this case, they stopped here. They didn't keep eating until they got to that point. So they've not overconsumed protein very much, but they've underconsumed carbohydrate a lot. The shape of that turned out to be incredibly important because we found that shape in other animals. Now, if you're a locust, you do best if you're at that point, and you suffer if you're in this region or in this region. You suffer in this region because you get obese and you die sooner. Granted, it's pretty tough to tell whether a locust is obese because it's got an exoskeleton. But inside, they're really chubby. It's a little bit like, I kind of like this analogy, it's a bit like having a medieval knight's suit of armour and being made to wear one which is several sizes too small. So you're really squashed and uncomfortable inside, and that has certain costs. So they suffer obesity. When you put them on a low protein diet, they overeat to get limiting protein and they end up obese. In this region here, they end up very lean. They use up all their fat supplies because they haven't eaten enough total energy, and they're susceptible to starvation. And that's a real cost if you're a locust in the wild. So, the summary of this shape is that protein intake is prioritized when the diet forces a trade-off between protein and carbohydrate. If you can get to that point, great. If you can't, then you're going to try and prioritize your intake of protein, no matter what that means for your total energy intake, if you're a locust. But also if you're a rat. So we did experiments on rats and mice and found the same pattern appearing. If you allowed them the opportunity to mix their diet with proper complementary foods, they would converge upon an intake target. If they couldn't get there, then they would maintain their protein intake more constant than their carbohydrate and indeed their fat intake. So you'll see that strikingly here. If they're on just too low a percent protein diet, they keep eating until they've got the right amount of protein relative to the target, and in so doing overconsume carbohydrate and hence calories, and hence become obese. We found that also in fish, we found it in chickens, we found it in monkeys, we found it in a whole range of different animals, all of them omnivores and herbivores. So that shape looks like it means something important in a general sense to omnivores and herbivores. Does it matter to us? Because we're an omnivore. Well, to answer that question, we did an experiment. We did an experiment with students, as one does, because they're cheap and enthusiastic. And disposable, yes, exactly. Their parents mind, but I'm a parent, I understand. So anyway, no, what we did was we took some Oxford undergraduates up into the Swiss Alps. And one of the other advantages with at least some Oxford undergraduates is that their parents are rich. So that meant that we had somebody who had a chalet in the Swiss Alps. And we were able to take a cohort of undergraduates and to incarcerate them in this chalet in the Swiss Alps. We then provided them with breakfast, morning tea, lunch, afternoon tea and dinner from a buffet at each occasion, from which individuals could take what they wanted, and we measured how much they ate, and from that were able to extract the equivalent plot to the one I've shown you already. Protein eaten against, in this case, it's non-protein energy intake, carbohydrate and fat combined. Now, it doesn't mean you always need to put those two together, but you'll see why in a minute. The main signal from the data comes if you do so. Non-protein energy versus protein energy eaten. So what we did was we initially allowed all our students to do what they wanted for several days and eat what they wanted, and we measured what they ate, and that was this point here, and we can presume that that's somewhere close to the intake target. We then confined some of them to lower percent protein food items and others to higher percent protein food items for a period of time, and then put them back onto the free choice. And just to cut that long story short, we ended up with, on the low protein period, the students overconsumed total energy and ended up with rather similar intakes of protein to during the free choice period, whereas when they were put onto the high protein dietary regime, then they ate less energy and didn't overconsume protein to any substantial degree, but instead ate quite a lot less non-protein energy. So just those 3 points is starting to be reminiscent of that pattern I showed you before for locusts and for rats. We then went backwards through the literature and found all the other studies where people had confined subjects to eat what they liked over various periods up to 6 months on different percent protein dietary regimes. These are those data just plotted on top of one another, unadulterated onto our plot. We've subsequently done a more recent assessment where there's now 23 different diet treatments, and they again form this pattern very, very strongly. So let's just plot that in a slightly different way. Let's take the slope, if you like, of each line as it intersects each point and say that's the percent protein in the daily diet against which we plot the amount of protein and non-protein energy eaten. These are the same points as here, but they're just presented in a different way. And it makes the point just as clearly, or more so, I think. What you'll see here is that the absolute intake of protein is remarkably constant across a whole range of different percent proteins in the diet, whereas fat and carbohydrate changes. The more you dilute protein, the more people ate. Okay? So it looks from those data But we too prioritise protein when the diet forces a trade-off between too much protein or too much protein or too little protein relative to fat and carbohydrate. One of the problems with this set of experiments, including ours, was that the subjects knew that the foods they were eating were what they were. And so there was the possibility that there were palatability differences, experiential effects, and so forth. And we needed some way of controlling for those. So what we did was we ran a trial here at the University of Sydney where we brought our subjects in to the Woolcock Sleep Centre, which is about a mile away from here, which is like a hotel, and we brought our subjects in for 3 full-week periods. During each week, they were allowed to eat whatever they wanted throughout 24 hours of the foods that we provided. We provided a whole abundance of different foods, every single one of which had been designed to be either 10, 15 or 25% protein. They were matched for their palatability, and individuals couldn't distinguish the 3 apart. And what happened was our subjects spent 1 week where everything they ate was 10% protein, 1 week where everything was 25% protein, and another week where everything was 15% protein. So far as they were concerned, they were getting this abundance of the same foods and similar menus on 3 different weeks, and they had no idea that they were different in this one key respect. So to give you a feel for it, that's the 10% protein lunch from one of the days in the trial, and there's the equivalent lunch from the 25% protein week. So you've got wraps, and you've got sushi rolls, and you've got apricot muffins, and you've got salady bits with low-fat dressings. And these taste different. These are sweet, they're savoury, but they're in fact, in every single case, within this group here, they're all the same nutritionally. These are all the same nutritionally, but they're 25 rather than 10% protein. And in that way, we could control for variety, for availability, for palatability, and we could focus in on that one variable, percent protein. That took 3 years to do as a big study, and I'm going to distill it down to 3 points on our graph. There they are. So, our subjects overconsumed total energy on the 10% protein diet even when we controlled everything else, which is very strong evidence supporting the idea that protein is leveraging total intake. Okay, I'm just going to bring that together now in a slide which is a graphical representation of the implications. Here we have protein intake and non-protein intake. Here we have that array, which I'm claiming, and I think we now have good evidence, is something akin to the human rule for balancing intake of these 2 nutrient dimensions when you can't get to here. So, what happens if the diet shifts towards containing a lower percent of protein? Well, what we would predict would happen is that protein appetite, because it's dominant, would drive overconsumption. And that would lead to excess energy intake, and hence predispose towards weight gain. Let's just show a simple piece of Euclidean geometry to give you a feel for the power of that effect. These data come from the USA. In 1960, the US population, which was very typical across the world, had a diet containing around about 14-15% protein of total energy. The rest was fat and carbohydrate. Between that period and 2000, the US diet shifted to containing 12.5% protein, which is a dilution of 1.5%. Which sounds trivial. It sounds like a really little amount. What does that matter? How can that possibly have caused the obesity epidemic? Well, maintaining protein intake constant, which is what the US population did over that period, required them to consume 14% extra total energy in the form of carbohydrate and fat. Now, that is a substantial increase, and what's happened in the entire human nutrition literature over those same 40, 50 years is that people have argued about whether carbohydrate or fat is the real culprit, ignoring the fact that protein may be playing a really important role. And the reason they've ignored it is because protein intake hasn't changed over that period. If it hasn't changed, it can't be responsible. What we said was the fact that it hasn't changed is why it's responsible, because it's being regulated more strongly than other nutrient dimensions. But it still leaves the question, why did that dilution happen? Why has there been a tendency for us in the modern nutritional environment to dilute our diet with carbohydrate and fat? Because if you think about it, we're now in an unprecedented period in human history in that we have a greater abundance and variety of foods than ever before. And if we were really a highly adapted animal, as a locust might be, we should be able to perfectly regulate to this point. No problems at all, but we don't. We're behaving as if our diet is diluted in protein. Well, there's a whole complex of reasons why this has happened. And some of them seem to stem back to our evolutionary ancestry. If you go back to the Paleolithic, where our modern physiology largely evolved, you'll find that we lived in an environment where we had a diet that contained probably about 25% protein, and simple sugars were incredibly rare and incredibly prized. So if you look at the extent to which people, even modern indigenous peoples, will go to get honey and collect honey in the wild, putting their lives at risk, in different places across the globe, you'll see the power of simple sugars. Also, our main sources of protein in the form of game meat were very lean. They had very low percentages of fat. So modern meat, farmed meat, is about 20% fat. Lean game is less than 5%. So there's been an ancestral predisposition to find simple sugars and fats very attractive. So that's been one reason why we're easily diverted in this direction. Another really important one, it transpires, is economics. Because the cost of protein to produce is very much greater than the cost of carbohydrate, particularly carbohydrate and fat as well. So there's a pressure on manufacturing industries and indeed food production industries when you're farming animals, whether that be aquaculture producing fish or whether it be animal husbandry producing cattle and sheep or whatever. There's a pressure to dilute protein in their diet and also in the processed foods that are provided for sale to humans. That, of course, puts an economic pressure also on consumers. And what you tend to find is that obesity these days is more a disease of the less affluent than of the affluent. If you look at social condition against obesity preponderance, there's a relationship. The poor, obesity is now more a disease of the poor than of the well-off, which used not to be the case. 200 years ago, it was the other way around. If you were obese, it was a display of your wealth. So there are economic pressures pushing us in this direction. There are ancestral predispositions to find these things attractive. And there's also a couple of tricks, which I'm not arguing that they're conscious, but they're certainly effective, which have been employed by the processed food industries. So, for example, the major cues associated with protein come from flavours such as umami flavours and salt. Savoury tastes in food are really good cues, just in the same way that being touched was a cue of being in a crowd. Those taste flavours are cues for protein in food. And you find those now coupled time and again with high fat and carbohydrate snack foods. So things like potato crisps are just fat and carbs, but they're associated with umami taste, so they're savoury. And what we found in our Sydney diet trial was that our subjects on the 10% protein diet ate more by snacking more between meals, particularly on the savoury flavoured foods. So there's another indication of how we could be diverted away from an optimal diet by our current environment. Now that's going to be made worse, of course, if there's a reduction in energy expenditure, because principally energy to fuel activity comes from this dimension rather than this one. And so if you do less exercise, you're effectively shifting your target downwards, which means that if you remain on this diet, you've got to eat even more to get to your same protein target, and the whole thing can go round and round in circles. Now, in contrast, if the diet starts to incorporate a higher than optimal percentage of protein, then people will end up eating less and potentially then losing weight. But, as an evolutionary biologist, you are left asking the question, why is it that we're so unwilling to overeat protein? Why doesn't this curve do this? It stops. It stops short. Individual humans or monkeys or locusts or mice or chickens or fish would rather eat less energy and hence lose weight, which may turn out to be good if you're trying to lose weight, rather than overconsume protein. Why? Now, as an evolutionary biologist, you must, in the back of your mind, be thinking, well, I would believe that millions of years of evolution have led to animals not doing that, because if they did overeat protein, there may have been some consequence which affected their reproductive success. Well, that's a testable prediction. You can go about answering that in various ways in experiments. And we did so. So I'm going to go now to another insect. This is Drosophila. This is the animal beloved of geneticists, and it's been one of the most important biological systems in the history of biology. We know as much as we know about genetics because of this fly as we do of any other organism. Anyway, what we did was not take account of the fact that we knew a lot about its genetics, which happens to be useful actually when you're trying to understand the molecular and genetic mechanisms. But we were mainly interested in this animal because it doesn't live very long, and if you're going to do lifetime reproductive success and fitness and longevity measures, then it's good if your animal doesn't live more than a couple of weeks, because otherwise you're around for a very long time. So what we did was say, okay, let's try and confine these animals to a whole range of different nutrient intakes and measure the consequences for them. So what we did was we provided flies, in fact we provided 1,000 individual flies with one of 28 different diets containing different concentrations and ratios of carbohydrate and protein, and we measured how much each fly drank throughout its entire life. Now they drink their food, and they drink it in this case from a 5 microliter pipette. Now that's a very small pipette, it's a small animal. So it'll stick its spongy proboscis up the tip of the pipette, drink the stuff, and the meniscus drops, and you can measure how much they've drunk. And then you can plot their intake of protein and carbohydrate and measure for every given intake for each fly what the costs were or the consequences were for that fly. So what we've got here are surfaces which plot the effect on the fly in terms of its lifespan and its reproductive success as a female, the number of eggs it laid throughout its life. Now these are heat maps that are a little bit like mountains that sit on top of nutrient intake space. They have their maximal elevation in the deep red, and they fall to their lower limits. So the sort of the base of the mountain is in deep blue. So what you'll see here is that lifespan and lifetime reproductive success, how long you lived and how many babies you had, have differently shaped landscapes with respect to nutrient intake. So if you're a fly, if you want to live longest, it's not the amount of calories you eat that matters, it's the proportion of protein to carbohydrate that matters. The higher that proportion, the shorter you live. Now, it falls again beyond that dotted line there, but the maximal longevity turned out to be a 1:16 ratio of protein to carbohydrate in your diet. If you increase the proportion, you live less long. But if you wanted to make the maximum number of babies, you had to have a diet containing a 1:4 ratio. So you would live less long, but you would be more fecund. You would have a greater progeny. Your contribution to the next generation would be more substantial. So that allows you then to say, well, if you give the flies free choice, what would they rather do? Live longer or have most babies? Now, Darwin would have predicted that they would have more babies. He was right. So they behave just like nutrient-seeking missiles, and they reach the summit of the lifetime reproduction surface. But the key point from this is that you do need protein for growth and reproduction, but too much makes you live less long and also produce too few babies if you're a fly. Does the same apply to we humans? Well, we don't know. One step towards answering whether it does or not is to go to a short-lived mammal model system, and the primary short-lived mammal model for human nutrition is the mouse. And we currently have an experiment which is running up to its completion this year, where we have 1,000 mice on one of 30 different diets, varying in protein, carbohydrate, and fat. And this surface is food intake, so the surface itself isn't necessarily of interest here. But the point is that we'll know the answer. Does a mammal care in terms of its lifespan and its reproductive success about the balance of nutrients in its diet? If it does, and all the indications are that it does, then that has further implications for us. And we, of course, are in an environment now where there's all sorts of different fad dietary regimes that people are imposing upon themselves. So we're also doing our own experiment, and time will tell what happens in terms of medical outcomes. All right, so I've taken you on a journey from North Africa to aging and obesity via cannibalism and tickling things on the legs with paintbrushes, and all of that has addressed the relationship between genes, phenotypes, and groups and populations. We need now to go to the next stage and to bring that all together. And I'm not going to do that now because we haven't done it. But what we have started is a program of research where we're able to take some of our key insights from nutrition, our nutritional geometry that I've shown you some results from, those agent-based models of collective movement that I showed you earlier, and also models of landscapes and how foods are distributed and produced in landscapes. And by putting those three together, we can set free simulated agents which have nutritional behaviour, which is linked, their state is linked to their nutritional responses, their feeding behaviour, their foraging, their immune status, their reproductive outcome. All of these things we know because we have measurements of them in real animal systems. And we can set such simulated agents free within worlds that we can manipulate and see the effects of different habitat structures, different assemblages of species, and so on, and see how these nutritional interactions ultimately fashion the way that ecosystems work. So on that note, I think I'll stop. Thank you. On that note, I think I'll stop. Thank you.